Good morning, everyone. I'm the product manager of Spatial Transcriptome in BGI. So today I'm gonna talk about introduction of the Spatial Transcriptome. So let's start now. So today I'm going to talk about in these 4 sections. So first I'm going to talk a little bit about Stereo-seq technologies, which also including the overview of spatial transcriptome technologies worldwide. And then I will go to details about the Stereo-seq products of BGI. And then I will go through the applications. And the last will be the FAQs of Stereo-seq. So first, in general, that spatial omics has the imagery style of the field of life science. It's changing our understanding of the complexity of life. Unlike our traditional omics research, spatial omics not only focus on the molecular composition within organism, but more importantly, it reveals the spatial distribution and the dynamic change of this molecular at the cellular and tissue levels. So from this perspective, biology research can be more accurately interpret the communication between cells, structure, function of the tissue, and understanding the mechanism of disease occurrence. So in the 2018, single cell transcript technology was named the breakthrough technology of the year by Science, and then the single cell multi-omics research is becoming a trend. Then the combination of the single cell research with spatial transcriptome has made the study of single cell in 3D levels as a new spot. In 2020, the Nature have named the spatial transcriptome technology was the technology of the year by the Nature method, sorry. So further confirming that this technology is tremendous development potentials. So as we know that from the research perspective, that generally our research can go through 3 main stage, basically on transcriptome. So first, we will the first will be the transcriptome study of boxed cells, which could provide average gene expression level of large number of cells. While this method play a significant role in advancing our understanding of these characteristics of cell population, but we could not get the information of the specific cell within the population. Then the single cell came out that we could identify the cell type and revealing the cellular heterogeneities, but the spatial information of the tissue is lost due to the enzymatic digestion during the preparation of cell suspension. And the last, for the transcriptome technologies, then we can pick up the information of the spatial information that further promoting the study to accurate gene expression of cell in their tissue context. Moreover, the combination of single cell transcriptomes and the spatial transcriptomes for joint research allows for study of gene expression at spatial location to be integrated with ultra high resolution and the single cell levels. So we know now we're understanding the spatial omics theory, but what can the spatial omics really can achieve? That the innovation of this spatial omics was not can be mainly reflect in the following aspect. First, that the spatial omics emphasize that the comprehensive study of spatial structure and function of the biological system, thereby revealing the biology process more deeply. So in the field of ecology and the environmental protection, spatial omics also can be used to study the spatial distribution of pollutants and their toxic effect that provides scientific support for the environmental conservation. Additionally, spatial omics integrates the technology and method from various disciplinary fields, providing strong support for some complex life science issues, such as in clinical medicine. Spatial omics can assist in diagnosing and treating disease for more accurately. So, for instance, by in situ detection of the patient's patient's gene expression, it can provide a basis of future personalized treatment. So there actually there are multiple technology that based on spatial omics on the market, but the core objective of spatial transcriptome is to link gene expression data with its spatial location within the tissue. So that the spatial transcriptome is not technology that come out overnight. It's actually have been accumulated of decades of scientific process. Initially, the scientists rely on the microscope to observe the structure of the cells and tissues. Although this really early method could provide basic information about the cell morphology and the tissue structure, but they're not significantly for understanding more complex process. So due to the importance of the transcript information and the experimental detection, the spatial transcriptome technology can be has developed rapidly, and now it's mainly divided into 4 catalog based on the different principles. The first, as we know, we can call it spatial in situ hybridization, and the second we also called spatial in situ sequencing, and the next will be the spatial in situ microdissection, and the last was probably the most well known is the sequencing based spatial transcriptome. So for the first two, we also can be cataloged as they are image-based spatial transcriptome, and for the last two, we can say they are sequencing-based spatial transcriptome. So for the image-based spatial transcriptome, we most known such as fish, moorfish, sickfish, etc. This technology use fluorescent probes designed for the specific RNA sequence to achieve their in situ detection of specific genes. This method, like fish or moorfish, they through complex encoding strategy have the capability to detect hundreds to thousands of the genes. The advantage of this method is that they have relatively high resolution, which allow the gene expression to be localized at the cellular and even subcellular levels. However, the limitation is that the numbers of the genes that can be detected is limited, and the operation is relatively complex. And for the last two that we catalog as sequencing-based technology, that most well known will be the nanostring DSP, 10x Visium, and our Stereo-seq. And this technology typically involving placing the tissue section on chips loaded with tens of thousands of unique DNA barcodes or capture genes hybridization through barcoded hybridization probes. And these probes can be labeled different RNA molecules. And after purifying and recovering these probes, the high throughput sequencing is used to read out genes expression, and the spatial barcodes information from different regions. So for this spatial transcriptome sequencing based still facing challenges such as the cost and the complexity of the data processing. So now many research groups that have developing many effective data analyzing tools and integrate spatial transcriptomes with other omics technology to demonstrate comprehensive and complete regulatory networks of different research fields. And here I also listed some technologies that are mostly major. We can see the major commercialized spatial transcriptome method that each of them have their own strengths, such as like nanostring DSP. They have much friendly with TMA that they can do probably can up to 100 different kind of tissue samples. But they only can do such as bulk RNA sequencing on each field of views. And overall, I also compare with 10x Visium HDs that they have relatively higher resolution than their previously version. But overall, Stereo-seq still have the highest resolution and the largest field of view and no limitations on the species. So in general, how does Stereo-seq works? So Stereo-seq was the technology independently developed by BGI, and it captures RNAs from tissue section by stereo chips and restore the spatial contents by utilizing the spatial barcode, also called the coordinator ID CID, and such it is established a solid research foundation for further understanding the relationship between gene expression, morphology of cells, and the local environment. And each features that on these chips, we have millions of this spot, and each spot have 200 or 220 nanometers in diameter, and between each spot we have 500 nanometer in distance. And so there we see we have 500 nanometer resolution in general, which probably the most highest resolution now in the market. So there's a video that shows how this our CID and our props was located on our spot. And here are some other, and here that on the market, I also compared that the other technologies with Stereo-seq that such as for the 10x Visium that it also have the spot from its previous version that each of his spot have 55 micrometer in diameters. So in each of their spot, it can cover 4 to 5 cells. But in Stereo-seq, our spot diameter is around 220 nanometers. So in total, there are 400 spots on the cells under one cells. So that's the around 600 times different between the technology's resolution. So for our analysis, Stereo-seq also using the binning, which here can shows that beam 4 is around 4 spot times 4 spot here, and beam 20 is about 20 spot times 20 spot, etc. So in one cells, we around will be have around 400 spot under one cells, and here is approximately the area that calculate as the binning. And in additionally, the large centimeter level of working area is another advantage of Stereo-seq, that we have flexible to suit for tissue that any size, which is from 1 centimeter square to 169 centimeter, which is 13 times to 13 times squares. And for this size of organ is probably could including most of human organs in this field of view. So after all this principle introduction, then I will go more deeply with our Stereo-seq product workflow. So here is generally the workflow of our Stereo-seq products. And it's starting with slicing and the QC. So the sample preparation, like the OCT and binding tissue, will be prepared by our clients. And we in our lab, we will do the slicing and the RNA qualification, quality QC, etc. And and after that, we will do the sample staining to ensure the morphology of the samples. And then we will do the permeabilization to pick up the optimal time for the further experiment permeabilization. And then we will do permeabilization and for the reverse transcriptome and to have the cDNA released and further for the library construction. Once the library construction passed the QC, we will move to the sequencing. And currently, we will produce around 1 to 3 G reads for each samples. And for the biometric analysis, basically we have these 3 major softwares to do our standard analysis, that we will do the image studio for the primarily image QC for our samples, and I will to do the SAW pipeline to localization the samples with their spatial informations and their gene expression informations. And then the stereo map, we can do some visualization method, that if you have specific area of the samples you want to do further analysis, we can use this software to do further or customized analysis. And then for the sample requirement, we recommended to perform the tissue bending within the 30 minutes upon the harvesting of the samples, and the sample size should not be exceed like 80% of the coverage of the chips. For example, if you pick the 1 centimeter to 1 centimeter times 1 centimeter chips, so we will suggest that your sample will not exist 0.9 centimeter times 0.9 centimeter this field. And then for the thickness, we suggest that the thickness will not exceed 2 centimeters, and we also suggest the thickness of the OCT samples have to be greater than 4 millimeters. And then the principle of the tissue samples, we will say avoid any tissue RNA degradation and maintain the original structure of the tissue. And we also suggest that do not have any effect that will affect the further or downstream experimental operations. And for the sample transportation, we suggest that after the embedding, you can seal the bag in the sample box and storage at -80 degrees. If you are not sending them immediately. And also ensure that the sample will send with the dry ice. The minimum requirement will be 5 kilogram for the transportation. And the other details, we strongly suggest to go through our sample information requirement documents. And here also some suggestion about our sample preparation. Here is our Stereo-seq chips. So you can see here we have one error region on each chips. So it's necessary to confirm the position and the thickness of your samples to place on these chips. And here are the some introduction of the sample usage here. So for the first section of RNA-QC, we will probably slide 20 to 25 slides of the samples, and each sample will be around 10 micrometer thickness. So the basic usage of the sample will be around 400 micrometers here. And then furthermore, for staining, permeabilizations, and etc., the first section will probably around 1,000 micrometers here, and then we will do the library construction for the further parts. So as I mentioned earlier, that we suggest at least your sample will be require 4 millimeters at minimum requirement. And for the QC, for the sample QC first, we will do RNA QC that require the ring value will be greater than 7, and we will do the staining, HE staining, to ensure that the tissue structure no crystal, no deformation, and no cracking on these samples. And then after all this, we probably will do the library construction. And for the standard delivery, we will do sequencing. Read length is about PE 50 plus 100. So for the read one, we will have the CID sequence and the MID sequence here with the poly-T on the RIS 1 section, and for the insert section, we will start from the RIS 2. So the turnaround time, because for the sample preparation and the permeabilization time will be variable, depends on the sample quality and etc. So here we only calculate the TAT for library construction sequencing and standard data analysis. So it's around 25 working days. And because the data output will quite large, it's around 1 G clean rates. So we strongly suggest that use hard disk to delivery for our data delivery. And for the data, for our data analysis, here are the comprehensive data analysis software, with also called SAW. It's a set of pipelines that bundle to position sequence related to their spatial location on the tissue section. Qualify spatial genes expression and visually present spatial expression distribution. And it's also have actually in these pipelines we have 13 essential pipelines as well as other supplementary tools for supporting various and analyzing contents as well. So here is listed different software, not software, sorry, the pipelines to do our standard analysis. And so far, and also as our published papers, that Stereo-seq is compatible with multiple sample types. For humans, there's listed sample tissue types that we have successfully tested. Also for the mouse, and other than that, we also have some plants and some insects and specific animals used by our Stereo-seq technologies. If your sample was not listed here, we will willing to try to process the samples. We do not have any, as I mentioned earlier, that we do not have any limitations on species. So we are welcoming all kinds of the species and the sample types. And here, I'm gonna introduce a little bit about our latest released products that our technology also compatible with FFPE examples. That for FFPE examples, we use the free props instead of poly-T props. That we have tested on human, animal, plants, even on microbiomes. And for FFP examples, now currently we accept FFP blocks and rows, and we can do the same slide with HE staining. As I said, because the props are free, the random props, so it can capture the long coding RNA and mRNA information on the same time, and also it's species free, so it also can capture the information of microbiome and host code detections, and it's also remain the high resolution of its features. And here are some demo data that we have been tested on tumor samples, because generally in previous study for bulk cell transcriptome, that we have issues with duplications during the sequencing. So here we can see the sequence, the duplication of our FFPE Stereo-seq is relatively low values, and here as the total reads that's generated by our sequencing. Because most of our analysis still using mRNA data, so here shows the data have been used for downstream analysis is not as high as we as probably expected. But for the gene types, the median gene types detected, we see it's comparable with our FF samples. Okay, so that's all the workflows for Stereo-seq and for FF sample and FFP samples. So now I'm gonna talk about a little bit more about our applications that our papers have been published in different fields. So far, Stereo-seq technology have been published papers up to more than 60 papers so far, and 40 of them was have the impact in factors greater than 10. So the first well-known paper was published in 2022 on Cell, and the name of the paper was Stereo-temporal transcriptomic atlas of mouse organogenesis using DNA numbable pattern array. And in this paper, the tissue was from mouse brain, embryo, olfactory bundles, and the olfactory bundles and the brain was from 12-week-old female mice, and the mouse embryo was at 8 different time slot of development. And the researchers used the Stereo-seq have established a mouse organogenesis stereo-temporal transcriptome atlas, also called a MOSTA. And the research also the atlas include 53 sections, which analyze the spatial temporal dynamics of the transcriptome across the entire embryo. It's from the 9.5 days to 16.5 days at 8 different time points during the development. Among them, 13 sections come from different cut of the same 16.5 days of embryo and construct a 3D atlas of the embryonic development near the end stage, and the analysis in this cluster was performed on the embryo of each section, and identifying the main tissue and organ that matches each time, such as skin, bones, muscles, heart, lungs, etc. And there are tissue-specific expression information. So in this study, they also identified the spatial heterogeneous of cell types distribution in mouse embryo tissues. At the single cell level, spatial transcriptome analysis of the embryo is crucial for understanding the dynamic changes in cell fate. So during the tissue development, the researchers performed image-based single-cell segmentation on the 16.5 days of the mouse embryo, and they filter out cells with low capture rates and obtain about 280 more than 281 thousands of cells with an average over 1,100 UMI, also called MIDs, and 529 genes per cell. So after the unsupervised clustering and the visualized of the cell, 25 main cell types was identified, including epithermal cells, smooth muscle cells, endothelial cells, macrophage, etc. And here on the right, the video shows that the midbrain was has many important sensational regulatory functions, such as vision, hearing, movement, etc. So compared to other function area of the brain, the secular heterogeneity and the spatial composition of the cells in the development midbrain have been barely being studied. So the researchers performed single-cell segmentation and clustering on the spatial transcript data of the midbrain from 12.5, 14.5, and 16.5 days of mouse embryo, and identified 11 types of the cell, including the RGC, neuron blast, glioblast, GABA neuron, also glue neurons, etc. So in conclusion, Stereo-seq is expected to be extended to more medical field, serving as a complement medical image and histopathology data in future clinical practice. And for the second articles that is also the world first spatial temporal secular atlas of axolotl brain. So in mammals, if the brain has been damaged, it have great challenge to recover due to the limited regenerative capacity. But axolotl exhibit strong organ regenerative capabilities, and their brain anatomy of axolotl bears some similarities to mammals. So from previous study that reviews that the EGC that is a type of gliocyte was equivalent to mammalian neuron stem cells and play an important role in brain regenerations. At present, only a few EGC's activation and brain regeneration signal pathway have been confirmed in such as fish, etc. These pathways also have a regulatory effect on brain development that's suggesting the brain regeneration maybe have similarity with the development. So in this research, the axolotl telecephalon samples was taken by different developmental stage. With here says stage 44, which first feeding after hatching, and stage 54, which completion of the forelimbs growth, and stage 57 was the completion of the hind limbs growth, and also have the juvenile, adult, and metamorphic stage. It also include regeneration stage at different days after injured, which including the control group, which is juvenile samples, and 2 days, 5 days, 10 days, 15 days, 20 days, 30 days, and 6 days, 60 days after the injury. The spatial structure of the development stage 57 and the regeneration stage after 15 days of the injury has the highly similarities. So the comparison of the developmental and the regenerative neurogenesis, the analysts review that the maturation of neurons in both development and the regeneration process follows a similar transaction trajectory. So the study identified that the telecephalon has have 3 subtypes of EGCs. It has identified 3 EGCs in adult axolotl cephalons, which called WNT EGCs, SFRP EGC, and the RIB EGC. With RIB EGC, it may maintain a stage of high proliferation. The analysis of early period of the regeneration and the wounded area also induced the REA EGCs to develop into IPC. Then in mature neuron stem cell, and finally mature neurons. This confirms that there are certain similarities in cell differentiation of axolotl telecephalon development and regeneration, but they are also has their own specific molecular regulatory pathways. And I may not go through with the cancer paper, and I will go straight to the plants. So for the plants, we know we have many opportunity and requirement on plants Stereo-seq from rural area. So it's also a large field of in transcriptomics, spatial omics. So here also the first paper of Stereo-seq have been published in 2022 on developmental cells. That in this study, the sample was from 6-week-old Arabidopsis leaves, and the researchers selected 26 Arabidopsis leaves from 4 different chips for integration and the construction of a foundational data set. In previously planned single-cell studies, some cell subtypes were often very difficult to distinguish them by single-cell sequencing due to the high similarity, similar molecule features, such as the upper and lower epidermal cells, the palisade, and the spongy cells. So the Stereo-seq can identify and annotate the cells in the sample based on the image information, also the single cell level of the spatial expression. By combining the spatial information with transcriptome data analysis, it is successfully distinguish between the expression similar upper and lower epidermal cells, as well as for the palisade and spongy cells. Also, further study the difference and the function have between these similar cell types. For explore the spatial difference in gene expression, the researchers has divided the leaves from center towards the end of 4 regions. So the zone 0 is the main vein, and the zone 3 is the leaf margins. So the result of the gene expression pattern shows that the photosynthesis gradually increase from the main vein to the leaf margins. While the main vein's primary role is related to the substance transports. Considering that the photosynthesis process is mainly occurred in the Mazervi cells, researchers further analyzes the gene present pattern in these cells, and the result was consistent with the previously analysis. So there are differences between the leaf vein and the leaf margin, but through pseudo-time analysis, found that the vascular cells are still unclear. This research, the cell in the main vein area is less differentiate compared to those leaves margin, and and the guard cells. Guard cells are significantly enriched in a branch of the development trajectory, but they do not show obvious spatial differences. So Stereo-seq still have huge potentials in plants research because of the specificity of the tissue and species. We welcome all kinds of opportunities, and the technology has now been covered all kinds of species and tissue types, but BGIs has a lot of experience on chromatography. So as long as the sample was embedded according to our sample requirement, we can have a try with different types of samples. So here, I would like to introduce a little bit about our grant project. Here is especially for plan science grant project that if you have submit your project plans to us, and if we have well, we will do the evaluation based on your project plans, and if you once you win the grant project, we will have 50% off of the products. So the deadline is before the end of this month. So you can contact our sales for further information. So here are some FAQs along with our last year experience with our projects. So here are the most top 5 frequently asked questions. So first will be what kinds of chips do we offer now? So currently we mostly offer 0.5 times 0.5 centimeters and 1 times 1 centimeter square chips for commercial delivery. But if you have the sample greater than this size, we can do the customized chips as well. But we need to do some evaluation before the project. And it is also, is it possible to do multiple samples on the same chip? It's experimentally durable, but we do not suggest this operation because if you have the samples with different tissues, then you probably may have different time for permeabilization. And if you did not pick the optimal time, it could affect your result greatly. And for the third question, it will be if the sample failed the first QC, could we repeat the QC on the same OCT samples? For this, we do not highly recommend it, but because as I said earlier, that for each OCT we have certain thickness requirement. So if you have already take the first QC, and if your sample thickness was not thick enough, and we may have not enough samples for the later experiment. So it's also durable, but not highly recommended. The fourth question will be how to avoid crystal formation. For January, we will say ensure the excess liquid on the tissue have been removed before embedding, and rapidly freezing the samples. But we do not recommend it for the nitrogen freezing because it may cause the sample explosion. But if it too slow to freezing the samples, will cause large crystal in the samples. And the last question will be for the permeabilization section. Is the longer time the better result? That is incorrect because for the long permeabilization time, it may be cause RNA diffusion. After completion of the tissue removal, it will have the same exposure setting. So we will say we will normally for the permeabilization time we will have 4 different time slot, and we will pick the most optimal time slot for our further experiment. So that's all for my topic today.